So targeted genome editing is the topic of our discussions, not only in this class, but in the upcoming sessions as well, because I want you to understand that as much as possible as a undergrad student about this special area of research or area of technology, I must say. You remember the things which were related to creation of transgenes, sending external genes to the mammalian genome, to the human genome, and to the genome of animals. When you send an external gene with that intention that it should become the part of the genome, it should impart characteristics to the genome, you are not able to know about certain important things about that activity, about that strategy. The first thing you do not know is that how many copies of the genes will be incorporated into the host genome. You never know that there will be one copy will be incorporated, there will be 10 copies will be incorporated, there will be 100 copies of the external gene will be incorporated into the genome. You never know that. So you will not be able to control actually that how much or how many external genes or the copies of the external genes should be incorporated into the host genome. That is a very important variable which you can't control in the creation of the transgenes, because you don't know where that gene will be landed. Think about the concept of the genome. It's a very big thing, very, very big thing. And you are sending an external gene and you don't know where it will land into the genome. It might be landed into some very important part of the cells, some very important part of the DNA. But theoretically, since we believe that 98%, and we know that 98% of the genome is like a protein, a non-coding part of the genome, so it will be landed into somewhere which is called as gene desert, where no gene exists. But there is a serious possibility of integration of such genes into some critically important places, and that might lead to some severe consequences during the incorporation of external genes. You should ask that question to yourself first. I have answered that question in earlier discussions, that what is the need of sending an external gene? What is the need of sending genes to human cells? And the requirement is that the reason is very simple, that we want to incorporate the properties of our own choice. We want to replace some faulty genes, or we want to add qualities by sending external genes into human genome. But we don't know how many copies will be incorporated. This is the first thing we don't know. The second important thing we don't know is that where that external gene will be incorporated, at what place within the genome. You know, it is like throwing a stone from the International Space Station to the Earth. It's very difficult for you to predict that where that stone will be landed on the Earth. Genome is that much big in perspective of the external gene. You never know. So you don't know where it is landed. So if, for example, in the future, if you want to manipulate human genome, if you want to act like a god, if you want to play with the creation of God, if you want to interfere with the natural evolution procedures, nature has evolved our genome over the period of millions of years, and we want to make changes in our genome, we need to be very precise in that action. We can't be like that. We don't know how many genes are incorporated, how many copies are incorporated. We can't be like that. We can't be like that where that external gene is landed, where that external gene is incorporated. We can't be like that. The human genome editing or human genome enhancement will only be effectively possible if we can control these two things. How many copies will be integrated and where that external gene will be incorporated. These two things like an important bottleneck which is resisting a successful technology for human genome manipulation. So we need to know, we need to have certain things. We need to be more precise in our experimentation, so we can tell that we able to incorporate that external gene at the place of our own choice. So that is from where the requirement of targeted genome editing came into existence. Before that, it was genome editing. We are just sending things, and then after testing, we just came to know the things are incorporated, the things are not incorporated. If they are incorporated, where they actually integrated, we don't know. In order to control that activity 100%, we should know how many copies will be there, will be incorporated, and then number two, what is the place where these external genes will be incorporated. If we have a control on that thing, that means we might be able to successfully edit the human genome. Otherwise, whatever you have studied from me about the transgene and about the editing of genome, those techniques are too risky to be used on humans. For example, if somebody believes I can replace the thalassemia gene in the expected baby of a couple, उनके expected baby में thalassemia faulty होएगा, I have calculated. I want to fix that error in the gene of that baby so that the baby can raise himself or herself in a normal way. It's a very good thought. It's very sweet. Therapy, ethically, morally, and ethically, it should be appreciated. But when you are manipulating the baby, when the baby was in a single cell stage, at the zygote level or at the stem cell level, when you are trying to engineer the genome of that baby, you are sending an external copy of a corrected gene. You will not be able to control where that external copy of a functional gene will go. Who knows? It might be ended or landed or integrated into a very important place in the genome. Who knows that multiple copies of that gene will be integrated into the baby's genome. So after all that laborious activity, when the baby will be born, with the multiple copies of that corrected gene, it might ended up into new troubles. We can't risk human lives for such experimentation until unless we have full command and control on that type of technology. That was the need. That was the requirement of targeted genome editing. I'm sure you understand the background that why we need to have that technology which can accurately incorporate the gene of our choice into mammalian genome. The start is very simple. The start was very simple. The end is very fancy. It's not the end yet. The current fancy state of that technique is called as, let me start from the end, is called as CRISPR-Cas9 system. So all you wonderful people out there, if we successfully manage to execute that lecture, we will start with the earlier, simpler ways of targeting the genome. And when we conclude our lecture, we will be studying about the CRISPR-Cas9 system. So the CRISPR-Cas9 system is the most accurate way of editing a genome right now. But teaching you the CRISPR-Cas9 system without explaining the reasons and the requirements behind is not the very best of the way. Or telling you about the CRISPR-Cas9 system without telling you the earlier technology is also not useful. So that is why we will see the growth and development of the techniques associated with targeted genome editing. You know, our kids, they know the smartphone only. Smartphone, that's all. That's all they know, smartphone. They have no idea about the Tetidas, the phone with the buttons. They have no idea the life before the cell phones, when people are dependent, on the landlines to communicate with each other. I don't know I should tell you on the YouTube that thing or not, but at that time, I usually admit all my faults in the classroom. And hum aisa karte the, hum aisa karte the. Such a beautiful, innocent signals people have created at those times. Because the landline, you never know who is speaking the call. So you need to be innovative. That only the target person should pick the call. So it was like, एक बार bell होएगी, फिर बंद कर देंगे, फिर दो बार फिर bell होएगी, फिर third time जो bell होगी ना वो मेरी होगी. Just pick the phone at that time. आज के context में those things are wonderful. Looks very innocent ways of, and then the life before the landline. So understanding the smartphone and this advanced version of technology, and might be wonderful if we know the history and growth and development of that technology here. So let me start our lecture slides. Actually, lecture has been started already with the comparison of different versions of information processing out there. So we have three forms of information which are quite comparable. You know about the letters, which when put together, they create the words, which create sentences, and it creates paragraphs, and then the books. And you can understand everything. You can understand everything if you have enough patience to read the book. I wish मेरे पास भी ये patience, so I'm not very good in that reading. 24, 25, something like that, or 26. I'm not. I can't remember the alphabets. Alphabets of English are out there. 24 to 26 alphabets of English out there. Different combination of those alphabets can create any sort of expression on the textbook. You can make any sentences. It's a giant leap. Binary code. Imagine only two codes, 01, 01, 01, used to create programs, and they are the basis of our computer technology. Whatever we are able to do with the computers today, whatever the things we are able to execute with the help of computers today, they actually originated from this binary code. Yes, no, yes, no, yes, no things. And what about the genetic code? What about this? 4 alphabets. Different combinations create different genes. And different genes, when put together, they make genome. They're making genome. And the genome is doing what? Humans. All versions of life you can see, the most complicated things you can ever see on the planet Earth are originated from where? Are controlled from where? Using simple 4 base pair, using simple 4 letters. Life is so simple for the God that it was written using just 4 alphabets. If it was written with the help of more than 4 alphabets, you can assume the level of complexity. I believe when the aliens come to our planet in the future, maybe they will think that there might be life on this planet a billion years ago, like we used to speak for the Mars. There might be water out there a million years ago, few million years ago. So the aliens will be talking about our planet. There might be life, and that life was so simple that it was made from the 4 alphabets. I saw another concept in another movie, sci-fi movies. I don't remember the name. The alien carrying a DNA with 3 strands. The level of complexity will be huge. If we able to do wonderful things, if we able to do all these great things with just 4 alphabets, think beyond the box, having a 5th alphabet. Think about having an extra strand of DNA. It will create amazing versions of life. Who knows? So the level of complexity in the genome and the comparison should be in your mind before thinking about editing it. The purpose and the agenda of the lecture and the topic is to get the knowledge that how we can edit the genome. So first you need to know about the different types of genome present in different species. When I talked about editing a genome of E. coli, a bacteria, it's about 4.6 million base pairs, mega base pairs. When I talked about manipulating or editing human genes, that means I am talking about 3.2 giga base pairs. The numbers are mind-boggling. I'm telling you the difference. It's not about the mega and giga. The difference between mega and giga is so much that every single zero added is not making a change. It's not making a change. It's making a hell of the change. So editing 4.6 million base pair is a peanut, is nothing as compared to editing the 3.2 giga base pairs of the human gene. Okay. So, and you know, we are different from each other, particularly the mammals, with a very small difference in our genome. For example, I should tell to Farhan or Hamza or Mustafa that you are almost 60% banana genetically, and we are different from chimpanzees because our genome is different for just about a little more than 1%. So we are humans just because of the difference in our genome of around 1% or so. That means the little change in the genomic architecture changes the outcome of the species at all. It actually defines who will survive and who will not. When dinosaurs were there, if we were there at that time, we might be thinking might is right. That is a law of nature. Dinosaurs are huge. That is why they were the masters. Dinosaurs were the humans of that time. They were like the masters of everything. Everything is defined by the dinosaurs. Who will live, who will not, defined by the dinosaurs. Consuming everything, everything is working for the dinosaurs. And then there was a famous hit by the asteroid. And that results, cloud, and then the sunshine was not there for few months, and then vegetation is not enough. So the might and the power, which was a law of nature at that time, proved to be a curse for the dinosaurs. And they were gone from the planet Earth. The same thing, which was supposed to be a reason of survival, proved a reason of extinction as well. Pata kaun bache aapko? There were small mammals, very small. They were our earlier ancestors. A small version of mammals who choose to live in hiding at that time because of dinosaurs. They able to survive in that time, and they eventually evolved into humans. And then evolution just find another way that might is not the very best of the way to survive as a master. What was the best way to survive as a master? Now we know. Intelligence. Who is the master on the planet? Humans. Why we are the masters? Because we are more intelligent than the other species. And we are believing on intelligence in the same way the dinosaurs were believing on the power at that time. Who knows, the intelligence might not be the best of the ways for the survival. Who knows? It might be a curse, I don't know. I'm just proposing a concept for brainstorming. That's something. Okay. And the opening introduction, Gigi Like ne farmaya ki Allah is the master. Thank you very much, Like, for reminding this thing. We all know that, and we believe it in that as well. So in order to understand all these opening understanding that why there was a need to manipulate the genome was there, what was the need, what was the desire behind that activity, and to know the things which we are about to manipulate, the genome. So if you read the sequence of a certain organism with little errors, it might not be the same organism. It might be another organism. Okay, so I think the time is up for the first session, and with that opening understanding, and when we will be back in the second session, we will be talking about the first technique, which was effectively used earlier time. It was considered as very best at that time. The time was not very far. I mean, I was talking about 2005, 6, 7, 8, 9, 10, 15 years ago. Was considered very best at that time for creating a change or for manipulation of human genome at that. We will be talking about that thing. Okay, so thank you very much. Any question? हम time इस्तेमाल कर सकते हैं the rest of the time for the session. Okay, if you have any question about whatever I told you, please speak. You are free. जी like, अगर आप कुछ कहना चाहते हैं, आप हमें शाबाश, please speak up if you have any question. And if the session is interrupted, then we will meet again in the next session. Anybody wants to share his or her thought on that concept? Like, you're done? Okay. किसी का माइक्रोफोन अनम्यूट नहीं हो रहा है? I'm the only one who is speaking. Okay. कोई brave नहीं है बोलने के लिए? Share your thoughts on that wonderful thing. Do not get scared of manipulating anything. You know, nature and the creator gave us wisdom to do what? To try whatever we can do. When I said whatever we can do, that means whatever we can do. If we believe, and we do believe, that there is a creator out there, we eventually reach to that place. We eventually find the reality with the experimental means. And there was a very famous saying of a very famous scientist that when you know little of science, it actually makes you atheist. But when you actually came to know more and more about science, it helps you to get close to the creator. And I will be happy if you have any comments on that thing and your thoughts. Ji Kadesh, Shabash, thank you very much for speaking.
सर जैसे हमने अभी डिस्कस किया कि ये जब हम जीन को इनकॉर्पोरेट करते हैं तो वो मोस्ट प्रोबेबली वहीं जाके रखती है जहाँ कोई और जीन नहीं है, मतलब हेट्रोक्रोमैटिन रीजन। तो अगर तो फॉर इंस्टेंस ये उस जगह लग जाए जहाँ दूसरी जीन है तो ये उसे अफेक्ट करेगी या ये तब क्या होगा?
देखो, पहली बात तो ये कि हेट्रोक्रोमैटिन वो जगह नहीं होती जहां पर कोई जीन नहीं होता। हेट्रोक्रोमैटिन वो जगह होती जहां पर जीन एक्सप्रेस नहीं होते हैं।
एक्सप्रेस नहीं होता, यस है।
नॉन कोडिंग मैंने बोला था, वो जगह जहां पर कोई जीन नहीं है, क्योंकि 98% उसी से बिलोंग करता है, इसलिए हम ये अस्यूम करते हैं कि ये मोस्टली वहीं पर जाएगा। ठीक है? After the opening discussion of having a requirement for a targeted action to execute the genetic manipulation at the place of our choice, we have developed a certain techniques, and I will be talking frankly 3 out of them. They acted like a stairs, and one technique actually ended up into another one, another one ended up into another one. I'm sharing the most famous of among all of them. But the CRISPR-Cas9 system, the last one, which is supposed to be the most advanced way of creating a targeted genome editing, was evolved separately. It was not evolved from the earlier technique. The principles and methodology was evolved from another area of life sciences. We will see this. So the first thing we will be studying is about the zinc finger nucleases. It's not only the word zinc finger, the word used was ZFN, the zinc finger nucleases. The second technique which we will see in the meanwhile was the TALENs, or transcription activator-like effector nucleases, also followed by nucleases. And CRISPR-Cas9 system were clustered regularly, interspaced, short, palindromic repeats. They also associate with the nucleus. And the name of the nucleus is Cas9. So the Cas9 is a nucleus, and you know all nucleases, they have the ability to cut the genome. So we will start with the first one, the zinc finger nucleases. Zinc finger proteins are naturally occurring DNA binding proteins in our system. Remember that our genome is just a arrangement of base pairs like a thread. This is what we have as a genome. So it should be so our genome is like that. Okay. that genome contains large number of genes. These genes can be regulated with the help of transcription factors, which binds to the place, which binds to a target place, and then activate or suppress the function of the genes. I used to speak that statement. It's not about having the genes, it's not about having the DNA, it's about how we can control and regulate the expression of those genes. Life is not having a certain base pair present in a certain sequence. It's about how we can regulate the expression of those sequences. And this is the job of the proteins called transcription factors. Just one example of the proteins. These proteins are supposed to go and bind to a target, a particular place. These proteins are supposed to bind at a particular place. Those are called as DNA binding proteins. They are regulatory proteins responsible for the regulation of genes. The DNA binding proteins traditionally have a specialized domain. Every DNA binding protein is made up of multiple domains, but one of the domain, one of the domain is created in such a way that it can sense or it can recognize one particular sequence of the DNA. So one part of that DNA binding domain always able to recognize a target sequence, a specific sequence of nucleotides. That is why that transcription factor or DNA binding protein is able to find a place on the genome with the help of the DNA binding proteins. There are different types of DNA binding proteins exist out there, and one of them is called as zinc finger proteins. So there are many DNA binding proteins out there, and I am just mentioning one of them, which is called as zinc finger proteins. Zinc finger proteins are also DNA binding proteins. Their special thing is that its DNA binding protein is able to recognize 3 base pair. It can recognize 3 base pairs at the same time. So every zinc finger protein can recognize 3 base pairs on the genome. Now we have large number of different types of zinc finger proteins in our system, but each zinc finger protein is able to recognize 3 certain base pairs. If we engineer these 3 zinc finger proteins in such a way, if we connect them together by genetically engineering means, if we connect them together, we will be able to recognize 9 base pairs of a target sequence. We can specifically target 9 base pair. If we put 4 zinc fingers together, we will be able to target a 12 base pair sequence on the genome. The approach is limited in this aspect that we are dependent on the choice of zinc fingers. That each zinc, for example, if this zinc finger is able to bind with GGG, it can only recognize 3 Gs in one sequence. So we need to pick different types of zinc finger proteins, put them together to make a zinc finger array of our own choice. It is not completed yet, since after creating a DNA binding domain of our own choice, we need a nuclease. We need a nuclease which should be able to create a double-stranded DNA break on the DNA. So now taken together, this structure is a nuclease. What that means? This structure is able to create a double-stranded DNA break because it contains a nuclease. The second special thing is that that structure contains a DNA binding domain, and that DNA binding domain is designed by humans. We can design it. In total, what we are able to achieve by creating or by designing zinc fingers together and by putting a nuclease on it, we will be able to create a nuclease which can go find and target the place of your own choice and can create the double-stranded DNA break at the place of your own choice. For this purpose, they have included the fork1. They have included the nuclease which is called as the fork1, this one, fork1. So fork1 is the nuclease which they have selected at that time for this work. And there is a very big reason why they have selected that. Because the ultimate purpose for designing any targeted genome editing technology is that whatever you are creating should be specific, should go at the place of your own choice. It should not create any off-site activity. Off-site activity means it should not go to any other place where you are not intended to. The reason why they selected the fork1, they selected the fork1 for that reason. The zinc finger nucleases work in pairs. As we know that the DNA is made up of double strands. The DNA are made of double strand. So this is one strand of DNA over there, and then there is another strand of DNA out there. We create two zinc finger nucleases which can target two different locations on different strands of DNA. So like this zinc finger nuclease has a sequence specificity to bind with that DNA at that place. And then we create another nuclease which can find a sequence specificity on the other strand at a certain distance. That distance is called as spacer or spacer sequence, which separate these two DNA binding regions. If we able to create this arrangement, we able to send two nucleases which binds on the opposite strands, providing a condition where these two nucleases, fork1 nucleases, came in front of each other. The special thing about the fork1 is that it cannot cleave the DNA from one side. It requires a dimeric structure. It requires two fork1 nucleases to be present at one place. So the two arms of the nucleases works. The fork1 will activate it once it is present in front of each other like this. Now imagine the level of specificity. If this sequence finds the right place, this sequence finds the right place, these two nucleases able to bind with a place of your choice, that will take these two fork1 nucleases in front of each other. They become activated and they will install and create a double-stranded DNA break or DSB. If either of them fail to bind at the target place, the fork1 will not be activated. But that means the double-stranded DNA break will not be executed. Now, in order to have this double-stranded DNA break here, imagine the things required to execute this activity. Number one, you need to have two nucleases. You should have find a place which should exist. You should have two nucleases having or separated by a spacer distance from each other so that the fork one can see each other and become activated to create a double-stranded DNA break. You need a sequence, you need a specific nuclease, you need a specific zinc finger array. After that, you should be able to successfully send these two nucleases within the cell. Agla limitation, you should be able to send both of them into the cell. If your experimental technology is not efficient, you might be able to send one of them, but not the other one. The next limitation, both of them should be there. The next limitation, both of the nucleases should be able to find their target sequences. And then, of course, the last thing, they should be able to create a double-stranded DNA break. So all these things are required in order to execute a double-stranded DNA break at the place of your choice. In order to increase the specificity, you can add more zinc finger to that array. Adding 4 zinc fingers means 12 base pair sequence. That is good enough, because a 12 base pair sequence is unique in the human genome. It will not be repeated. If you add one more nuclease to that system, one more zinc finger, that will add more specificity. Okay, increase it. So these are the limitations associated with this function. After having a double-stranded DNA break, the next procedure is no longer dependent on human activity. So we use our natural DNA repair mechanism, the cellular DNA repair mechanism, to conclude the story. So think about a double-stranded DNA break is created as a result of the zinc finger nucleases. After that, there are two choices for our cellular defense mechanism, for our cellular DNA repair mechanism to fix this double-stranded DNA. You need to ask one question to yourself here, that is it natural of having a double-stranded DNA break in our genome? The answer is yes. Because of injuries, because of dysfunctional activities of the protein, because of XYZ reasons, and particularly during the synthesis of DNA, when we are creating the DNA, it happens that a certain portion of the DNA was missed because of those things. So there is a need of a full-scale mechanism which should be able to fix the double-stranded DNA, which should be able to fix the double-stranded DNA. And that mechanism is called as our DNA repair mechanism. Once the DNA repair mechanism finds a double-stranded DNA break, it proceeds in two mechanisms. One of the mechanism is called as non-homologous end joining. Is called as non-homologous end joining. That means if the DNA repair mechanism does not find any external DNA at that time, the cell will not allow that double-stranded DNA to exist. The cell will fix them together. The cell will try to connect these double-stranded DNA to restore the genetical integrity. For that, the first thing the cell will do is to capture the broken or separated double-stranded DNA and then try to connect it. During that process, the DNA repair mechanism might be incorporated some base pairs. or the DNA repair mechanism might be deleting some base pairs in order to put them together. Either when they are repairing, like if there is a breakdown in the wall, and you ask somebody to fix that broken part of your wall in the house, the person might delete some more bricks in order before fixing it perfectly. So the nature choose either to add some base pair to join it, or to delete base pair first to put them together. In both the cases, the natural sequence of that place will be gone. Adding one base pair is not nice. Adding multiple base pair is not nice. Removing few base pair is not nice. So whatever is happening, adding or removing base pair will ended up in a new DNA sequence which is not equivalent to the wild type. So what that means, if we able to successfully execute the zinc finger based nuclease activity, that will create a double stranded DNA break, followed by the DNA repair mechanism called non-homologous end joining, ended up in a repair which leaves an error. What that means, the error, there might have be more base pairs out there which should not be there. They are added during the repair, or there might be less number of base pair out there which were deleted while repairing. In both cases, that place lost its integrity. Assume that place is an exon of a gene. Assume that place is an exon of your choice, because you can design the zinc finger nucleases by yourself. So you can design the target, you can send it at the place of your own choice. So you can create a double-stranded DNA break on the exon of some gene of your own choice. After that non-homologous end joining, the resultant gene will have a different sequence than the wild type. In easier word, that means the exon will lose its functionality because the sequence is changed now. In more easier term, that means it will not going to produce any wild type protein, or the function of the gene is gone. And this is what we call as creating a mutation at the place of our own choice, and it is called targeted mutation. And that was supposed to be an excellent technique to destroy the function of a gene of your own choice. अपनी मर्जी का जीन मुंतखब करें, design zinc finger nucleases and destroy the function of that gene. So one, and that when that technique was there, it was applauded a lot, because for the very first time we able to send the things at the place of our own choice within the genome, and that was amazing of that time. The other way is a little bit more laborious as well as more complicated. First, creating a double-stranded DNA break, and then using that double-stranded DNA break to incorporate the gene of your own choice, to incorporate an external piece of DNA. So the first thing is that creating a double-stranded DNA break at the place of your own choice. Look at that side of the slide. We are sending, along with the zinc fingers vectors, we are sending other donor plasmids as well. The zinc finger nucleases will enter into the cell. It will create nucleases, two nucleases, which will find the target site of your choice. It will go there and then it cleaves the double-stranded DNA. Full stop. Now, if you are not doing anything, what will happen? If you are not doing anything, this will happen. Non-homologous end joining. But if you intervene at that time, you are sending another plasmid, donor plasmid at that time. Look at the color coding of that donor plasmid. The HAR is here, the HAL is here. R means right side and L means the left side. So the HAR, which is in blue color, this has the same sequence of that part of the DNA, the blue one. The red side, the left side, has the same sequence of DNA similar to that left side of the target place. The DNA double-stranded break is supposed to happen here. And what will happen? That after the double-stranded DNA break, the DNA repair mechanism will rush to the site and will start repairing the function. But if you provide that donor DNA, if that donor DNA is there, since it matches with the open ends, can you see? If the double-stranded DNA is, if the double-stranded break is here, this part matches this side and this part is matches this side. So the DNA repair mechanism utilize this external DNA and incorporate it in order to fix the problem. What was the problem? The problem was that double-stranded DNA break is there. The DNA repair mechanism will try to use that external donor DNA to fix or to connect that double-stranded DNA break. And what will be the result? The result will be a new DNA with matches the sequence, but along with the yellow part of the DNA which you have created. So in summary, when a zinc finger nuclease is assisted by a donor DNA with the same sequence, you can send that yellow DNA as well at the place of your own choice. Now imagine or assume that the yellow part of the DNA is a GFP protein. If this is a GFP protein, what will happen after the double-stranded DNA break? Along with that donor DNA, the GFP protein is incorporated at the place of your own choice. And that was the first experiment where scientists were very happy, since they're able to send a particular gene, an external gene, at the place of your own choice. I want you to think about the level of complexities, the intermediate step of this successful result. This is a successful result, and I can tell you, I have done those experiments. मैंने ये किए हैं अपने MS में. My master's thesis was on targeted genome editing, and at that time CRISPR-Cas9 was not there, so I was working on zinc finger and talens at that time. This is a successful product, but this successful product is created as a result of so many variables. The variable starts from where? The variable starts from here. You need to design a target place. You need to create two nucleases. You need to send them at the same time. And now the complexity level is huge. Since you are not only supposed to send the zinc finger nucleases, you also supposed to add a donor DNA at the same time. What if one of the zinc finger is not working? The system will be crashed. What if the spacer distance is not good enough for the fork one to execute the double-stranded DNA break? The system will be crashed. What if the two zinc finger nucleases manage to be there, but the donor plasmid is not there? The cell is not able to collect all of them together. Putting all these things in perspective, it is highly less likely to put all these variables at one place to execute to get that product of your own choice. That is why it was so laborious that the efficiency level was less than, like, it's like 2 or 3% efficiency of that experiment. The scientist needs to work very hard in order to create a change at the place of their own choice. And that is because of the what we call as the zinc finger nucleases. But they able to do a lot of things, and most of the experiments was done in other mammals, including mice. They able to look at the time and the level of efficiency. The zinc finger assembly time is 6 weeks to create those nucleases. The injections on the 7th week, injections in the single cell to create the double-stranded DNA break, as well as if you do not add any donor plasmid, remember, you will end up in mutations at the place of your, that was quite easier. That was quite easier. But creating a double-stranded DNA break and then incorporating the external gene, that is little bit like you need some sort of luck in order to execute that experiment. And after that, you need to scan the region to see what is the sequence now after the manipulation and after the editing. So after that, they raise the babies, 15 to 58 pups. They develop 15 develop mutations out of 58. They identify using the PCR sequence, the target sequence. Look at that. This is the wild type sequence, the red and then the black one. The black one is a spacer sequence. Now you can guess the spacer sequence is the place where the double-stranded DNA break will be executed. And the DNA repair mechanism will try to fix it. Here, the DNA repair mechanism try to fix in this sample, and they deletes that particular portion, creating a mutation. Here, another mutation, here another. So it's a random option, but a mutation is created. What will be the next level? The next level is to incorporate the gene of your choice at that place. It's just creating a mutation. That is quite easier. So that is how zinc finger works in dimers. That we have a right side of zinc finger nuclease. We need a left side, design a zinc finger nuclease for the left side. If they're able to bind their places perfectly, and they're called as zinc finger because they do carry a domain associated with zinc. They're able to execute that experiment. Okay, so that is about the zinc finger nucleases. But when I was working on the zinc finger nucleases in 2010, that paper came which actually shows another type of DNA binding proteins which are called as talens. It was discovered from the plant sciences, and then they started using those proteins to create the zinc finger-like nucleases. And I will be talking on that thing in the next lecture. So I'm concluding my talk here, along with the belief that I have explained that zinc finger nuclease in its function. And you can ask your questions before the times run out. So I will be able to answer your questions related to that technique. Okay, so you can now speak or you can write as well. And if the class is interrupted because of the session, we will see, and I will try to answer the other questions on next class on Monday. Okay, so you are free to ask your questions. Please proceed. Anybody who wants to ask a question, koi unmute nahi hua. If you want to ask, if you do not want to speak, you can ask on the chat as well, but that is fine to speak. If you do not have any question, I should be very happy with my talk, that you have understood. If you understand the background and the need, that was amazing. And if you understand how zinc finger was working and the level of trouble, because whenever you read the papers, whenever you study the research material, you came to know, you will see the data and you will just bang your head in the wall ke itni effort ke baad itne se results aaye hain. Why? Always you ask yourself that it was such a laborious work, ended up in like nothing. So you might be able to answer that question if you understand the complexities associated with that thing. So Laiba asked a question, sir. Basically, we are using zinc fingers for the knockout purpose. if we are not sending any donor DNA, it will only create a mutation which you can call as a knockout mouse. So it was used to create knockout mouse, but also if you're able to send an associated donor DNA, you can send the gene of your choice as well. You can create a transgene as well. But more studies shows that it was successfully used in many experiments for the knockouts. Actually, that it was like a prestigious way of creating knockouts for many years, and then it was replaced by talens, and now we are no longer dependent on these two anymore. अब इसके ऊपर काम करना ऐसे ही है कि less efficient है. It's quite less efficient to work. It's quite expensive now to work on using, because we have more advanced and more easier way to manipulate the gene. But at that time it was like too much. लोग इससे बहुत प्रेस्टीज लेते थे जो लोग इस पे काम करते थे टारगेटेड जीनोम एडिटिंग पे. आई मीन कि हमें जगह बताओ, हम उसको म्यूटेट कर देंगे किसी में मैमेलियन सेल के अंदर. I have another question from Farhan. Farhan पूछता नहीं है, लिखता है, can other DNA binding proteins been used in targeted genome editing? Other proteins, actually the zinc finger nucleases or the zinc finger proteins was used since they have so many options. They are giving so many options to target so many different places on the genome. So like, and we can put them together to make like a target sequence of our own choice as well. So it is one of the best DNA binding protein which can be used. That is why it was used. The other DNA binding proteins can also be used, but they are not giving that many options like zinc fingers. And remember, if you can remember the famous restriction enzymes, the most famous word, restriction enzymes, XBA1, BAMH1, EcoR1, all those restriction enzymes are like this. They have two domains. The one is the DNA binding domain through which they can capture the DNA, and the other is the nuclease domain which is used to clip or to break the double-stranded DNA. ठीक हो गया? शाबाश. Questions? पता लगा हम मेडिकल बायोटेक्नोलॉजी पढ़े हैं, टारगेटेड जीनोम एडिटिंग में. Okay, shabash. Don't worry if the question is stupid, okay? Agar just write it to me. Let me, or if you can ask me not to speak your name as well, if you want, that is fine. I want you to ask questions, but you are no longer, you're not only limited. Please do not stay limited to that time only. If a question came to your mind about that topic later on, just ask me in the next class, okay? We will discuss it.